Hi everyone. Today's is the fourth lecture in the Chemical Oceanography module, and we're going to be talking about nutrients. So the issue with nutrients, and we're going to continue this when we talk about primary productivity later in the module, it all comes down to what exactly does life need to survive and thrive in the ocean? And why are some parts of the ocean really vibrant, filled with lots of animals, lots of plants, lots of stuff, lots happening, and why are some other parts of the ocean essentially deserts where there's not a lot growing at all? And this is a really complicated figure, but this is sort of the end goal of what we're building towards. And I want you to notice first a couple of things. So now that we're talking about nutrients and life and productivity, you can see we've really moved away from the deep ocean. We're looking at about the upper 300 meters. And that is because that ultimately, for the most part, hydrothermal vents excluded, sunlight is the ultimate energy source for pretty much all life on Earth. Except that this light is absorbed and scattered in a way that not a ton of it penetrates. You can see this yellow line here shows light penetration, so 100% at the surface. And by the time you hit 100 meters, you're already at 99% of the light has been lost and only about 1% of it penetrates. And so a lot of what controls the ability of things to grow in the surface ocean is driven by this light or this lack of light. So photosynthesis, which by definition, the photo and photosynthesis refers to light, is restricted to the upper part of the ocean where there's light available. So we've gone from 300 meters to now already we're just below 100 meters on average, where below this 100 meters you're limited by light. And you can see in yellow there it says light limitation. The other thing about what happens with light hitting the surface ocean is this sunlight is warming the surface ocean. And so if you look now at the orange line, this is your temperature, and you can see here your thermocline as identified there. And then at the surface, you've got your mixed layer. And so this warmth, the warming of these waters here are making them less dense. And so it's what we call stratification. So they're actually somewhat separated from these deeper waters below the thermocline. There's a restriction in the physics that allows them to mix. So this warm water is more buoyant than the cold water. And so this warm water, this mixed layer, sort of floats on the denser deep ocean. And the thermocline separates these two. And so in order to get mixing across this, you need some sort of wind or something like that. And this is actually important because light allows photosynthesis to happen, and it also makes this mixed layer more buoyant. So what this means is that if you're a phytoplankton living in the mixed layer, that's good because you won't be mixed below the mixed layer, if that makes sense. You're not just going to sink all the way out to the bottom because this is like stratified. It keeps you nice and safe up in the surface ocean and keeps you in the well-lit part of the ocean so that you can go ahead and do the photosynthesis that you need to do. Okay, so the other thing, so in addition to needing light and being sort of physically able to stay in the surface ocean, the phytoplankton or the plants in the surface ocean need nutrients. And here you can see in purple the concentration of nitrogen with depth. And so the concentration is higher at the bottom here, and you can see it goes down, 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 down. And right about at that 1% light zone, you can see the concentration basically goes to zero. This is the zero down here. And that's because anything that is living in the surface ocean, in this mixed layer with light, is going to immediately, as soon as it encounters this nitrogen, it's going to use it to photosynthesize. Okay? And so the reason it's gone isn't because it's not important. It's gone because it's super important, and it's immediately consumed. And that's why you see nutrients usually very low in concentration at the surface. Well, as a result, when you look at a vertical profile of something like particulate organic carbon, which is a way that we measure phytoplankton, you can see that they're much higher in this surface layer. And so this is everything that's grown on the light and the nitrate that was available. And so you see evidence that that nitrate was taken up and it was converted to phytoplankton biomass. You also see that here with the chlorophyll. in that the chlorophyll max, which is labeled as the DCM here, the deep chlorophyll maximum, is basically exactly in that place where the light and the nutrients come together. Okay, so this is a complicated figure, but it sort of shows you how everything is playing together and interacting to make this surface layer of the ocean a place for lots of biological activity. So phytoplankton require inorganic nutrients, and you can see here this is the sort of average composition of phytoplankton biomass. And they need a variety of macronutrients and micronutrients. This table has been completely, don't look at this, completely messed up. No, actually, let me not say don't look at this. Let me actually fix it. Okay, so the macronutrients are nitrogen, and phosphorus and silica are macronutrients, means you need a lot of them. And then the micronutrients are trace metals and vitamins. So that means you need just a little bit of them. This lecture, we're talking about macronutrients, so nitrogen, phosphorus, and silica. So you can sort of forget about the other two for now. And so now here we're looking at depth profiles. So this is depth here in meters on the y-axis for phosphate, nitrate, and silicate. And you can see their depth profiles are essentially identical, and that's because they're all macronutrients. And so in the surface ocean, way up here in the 0 to 150 meters, basically the concentrations all go to 0. And that's because that's that thin strip of the surface ocean I was just showing you where all of the growth is happening. So these nutrients are consumed to 0. But then you see something very different happens when you get to about 1,000 meters for all of these. The concentrations are at their maximum. Silica is a little bit lower, but the concentrations are essentially at a maximum. And what happens is that particular organic carbon or that phytoplankton that you were seeing in the surface ocean, it dies and starts to sink. So it mixes below that mixed layer and starts to sink. And that's where respiration starts to happen. Bacteria start to eat at it. And what ends up happening is, so the phytoplankton used up the nitrogen and the phosphorus to make this biomass, right? Which is why you don't see it in the surface ocean. And then they start to get chomped away at by bacteria. And they get chomped away at, and you end up releasing the inorganic nitrate and phosphate back. And that's what you're seeing here accumulating in the deep ocean. And so anything that has this kind of depth profile, it's called a nutrient-like distribution. And this usually means that it's something that's being consumed by photosynthesis in the surface. And then as the sinking organic matter is oxidized, it re-releases that nitrate, phosphate, and silicate back into the water, and you end up with these much higher concentrations at depth. And in these deep waters, there's no light and no photosynthesis, and so there's no mechanism to sort of take these nutrients out of the water, and so they just start to accumulate and accumulate. And so nutrients generally include the macronutrients of phosphorus and silicate, but really nitrogen is considered to be the sort of ultimate limiting nutrient. And so it's worth spending a couple of minutes explaining a bit about nitrogen cycling. So a nutrient, this is by definition, this is by an ocean definition, is a chemical needed to support photosynthesis and primary production. And here you can see a schematic. So this is N2 gas in the atmosphere, which in the dissolved gas lecture we learned there's a lot of it, right? 78% of the atmosphere is made up of N2 gas. And this does indeed dissolve into the ocean. And so there's some dissolved N2 gas in the ocean, and this is controlled by the Henry's Law, right, as we know about. But this dissolved N2 gas can't be used as a nutrient, which really, really sucks for the phytoplankton in the ocean. There's a handful of bacterial species that can do nitrogen fixation, which is this process where you take the N2 gas, the useless N2 gas, and convert it to ammonium, which is useful. But not a lot of things can do it, and you have to be in really warm, really calm surface waters. So unfortunately, that's not really a great option. So most of the ocean's food webs rely on ammonium and nitrate for growth, and you can see here. So phytoplankton take up ammonium, they take up nitrate, and then they grow, and then they die and get decomposed, and they release the ammonium back. Okay, and then the ammonium can be nitrified. So I don't know why they've moved this. This is the same ammonium as this. So the ammonium then is oxidized in the ocean because the ocean's an oxic environment to nitrite and then to nitrate. And then the phytoplankton can use the nitrate again, and then the phytoplankton die and they release ammonium. And then some of that ammonium is taken up, but then some of it's oxidized to nitrate and then to nitrate. And you can see you just keep going around this cycle. But the nitrate doesn't build up forever in the ocean. It is eventually removed. And there's a group of bacteria, when there's not a lot of oxygen around, instead of using oxygen to respire organic matter, they'll actually use nitrate to respire organic matter. And that's called denitrification. And they will take the nitrates and they'll convert it back to N2, which is now they've completely removed it from the usable pool and stuck it back out here to go with the atmosphere. So that's like a super short 5-minute introduction to the nitrogen cycle. But a couple of highlights are that the surface ocean organisms, for the most part, cannot use the inert N2 gas in the atmosphere, and they rely on ammonium and nitrate. And the other important thing is that ammonium and nitrate are used by phytoplankton, but then the ammonium is regenerated through decomposition, which is then oxidized to nitrate. And so everything is sort of cycling around. Okay, so if you look at the top left here, this is a view of the surface ocean nitrate concentrations, and you can see, as you would expect for a nutrient, since I just told you, nutrients go to 0 concentration at the surface because they're consumed. The bulk of the surface ocean is this purple color, which is basically there's no nitrate in the surface ocean, okay? But of course now you can see that that's not true everywhere. And so it gets at this idea of what's limiting growth. So for the bulk of the ocean, nitrate is limiting growth because you can see here the nitrate goes to 0, which means they have everything else they need, but then they run out of nitrate. And so nothing can grow and your nitrate's at 0. In some regions of the ocean, however, there's lots of nitrate, Southern Ocean being one example, and this upwelling region here being another, and the subarctic North Pacific. And what it means there is that there must be something else that's limiting growth because they have plenty of nitrate, so no one should be complaining. And indeed, you have to extend your definition of nutrients in this case. So in the Southern Ocean, for example, there's lots of nitrates. There's usually light, although sometimes they run out of light in winter. But what they're missing in the Southern Ocean is iron. And so you can see this high nitrate concentrations are because the phytoplankton have run out of iron, which is something else that they need. And this is known as the idea of the law of the minimum, that there's always something that's going to be run out first. And so the schematic of nitrogen in that little textbook chapter I posted for you guys has the values are really funky, and I don't know why. But typically, a nutrient-like profile is one where you have consumption controlling this really low concentration in the surface and remineralization leading to higher concentrations at depth. And nutrients, we refer to them as non-conservative because they have this dynamic behavior through the water column. So the concentrations of nutrients vary regionally, they vary seasonally, and they vary with depth. This is another look at vertical nutrient profiles. These are super classic old school ones. And the interesting thing about these is they show you this is phosphate here and nitrate here on the right side and the left side. And they show you that in the different ocean basins. So the Atlantic, Pacific, and Indian all have nutrient-like profiles for nitrate and phosphate, as you'd expect. But consistently, the Atlantic is always lower in concentration than the Pacific and Indian. And that's true for phosphate, it's true for nitrate, it's true for all of the various profiles. And the reason for this will become more apparent as you start to do thermohaline circulation. So the waters in the Atlantic are actually younger than the waters in the Pacific. And so organic matter is sinking and being remineralized and leading to this increase in phosphate concentration and nitrate concentration. But these waters in the deep Atlantic keep moving and they eventually end up in the deep Pacific. And so then there's more organic matter raining down on them, leading to more phosphate being remineralized and more nitrite being remineralized. And then with thermohaline circulation, it keeps going until it hits the deep Indian here. And so you can see that it's sort of this, the amount of nutrients in the deep ocean are a sign of how aged the waters are, that they've been just going around the deep ocean collecting all this organic matter raining down on them. It's being respired, and then you're reforming these inorganic nutrients in the deep waters. Now what's interesting about this chart is if you think about all the organic matter that's raining down, there's some implications of that. So you're converting this organic matter back to phosphate and nitrate and silicate, and you're doing it through aerobic respiration. So you can see, if you look at the oxygen profile in red, there's oxygen at the surface, it's equilibrating, it's produced from photosynthesis. But then at the same place where the nutrients start to regenerate, where they start to increase, the oxygen starts to go down. And you end up with this oxygen minimum where you end up with that maximum there. And that's because all this oxygen is being consumed to do respiration. And so that's why your oxygen values are much lower in the deep ocean than they are in other parts of the ocean. The other thing, of course, that goes along with that is when you dissolve organic matter or when you respire organic matter, remember that in addition to releasing oxygen during photosynthesis, you use up CO2. And so when you respire the organic matter, you're also releasing CO2, which is driving the pH down. And so you can see that as well, what the signal is. And so these nutrient profiles, or the sort of vertical profiles of things in the ocean, are really heavily controlled by what phytoplankton are doing in the surface ocean. So even though they're maybe only living and photosynthesizing in the surface ocean, their death and remineralization are controlling the profiles of all sorts of things throughout the deep ocean. So this is another view of the nitrogen cycle that I wanted to share with you guys. So the processes I've been talking about have been very general so far. I haven't been really highlighting a specific part of the ocean. But there are some differences between the open ocean, which you see on the left, and the coastal ocean. And that has to do with the amount of upwelling and the amount of nutrients that are available. So remember in the deep water, so here's your vertical profile of nutrients in purple and light in brown or orange, whatever this is. And so on the coastal areas, this deep water comes to the surface. And so it brings with it a burst of nutrients that can be used. The other thing that happens in coastal oceans is you have riverine inputs. And so your coastal ocean, you have the opportunity, you can see how thick this green layer here is that they've put in. There's a lot of growth that happens. You've got plenty of light, you've got lots of nutrient inputs and lots of growth. And so you've got lots of organic matter, this big circle here. But of course, that organic matter is going to start to be remineralized, and it starts to consume oxygen really quickly. And these waters can end up what they call being an oxygen minimum zone. So the organic matter is respired, it's releasing ammonium, it can be oxidized to nitrite, nitrate. But there's also, because you're low oxygen, eventually you can even run out of oxygen, and what happens then is denitrification happens in these coastal areas. And that's quite distinct from if you look over here to the open ocean, where you maybe have some N2 fixation, and you maybe have some mixing from these mid-depth waters up to bring some nitrate, but not nearly the way you have this sort of upwelling. A little bit of mixing up. And so you have some growth, but it tends to be this area where you've got some nutrients and some light, which is why it's sort of a little bit deeper here. And so you've got some organic matter that can grow and then be remineralized. And you see these cycles are all much smaller in the open ocean than they are in the coastal ocean. Lastly, I want to introduce the idea of the Redfield ratio. So this is something that's really interesting. There's this, a bunch of measurements were taken a long time ago, in part by this guy, Alfred Redfield. And what they found was that if you think about what's needed for photosynthesis, right? So you need CO2, you need water, you need nitrate and phosphate. We've already talked about that. And you form this organic matter. So when you measure phytoplankton, you measure this organic matter, the ratio of nitrogen to phosphorus is pretty constant around the ocean. It's 16 to 1, pretty much all over the place. And in fact, the carbon to nitrogen to phosphorus ratio is 106 to 16 to 1. And what's really interesting about that is when you then measure the inorganic nitrate, phosphate, and carbon, they also have ratios of 106 to 16 to 1 in the water column. So the concentrations of these species in the ocean, their relative concentrations to one another, are being controlled by the composition of phytoplankton. And that's basically the average. So like carbohydrates have a different composition than proteins, but if you smush everything together and average it, it's about 106 to 16 to 1. And so this is what you measure if you go out and measure phytoplankton, but it's also what you measure if you go out and measure nitrate and phosphate just as dissolved in organic species. And that's what you're looking at here. This is nitrogen concentrations as a function of phosphorus. And this line is the 16:1 line. And you can see this was the original Redfield plot. Now we've got much more sophisticated ways of looking at this. But for the most part, data from all over the world's ocean fall along this line of 16:1. And when you guys get into marine systems next term, you'll rely on this Redfield ratio a lot for understanding the biogeochemistry of the surface ocean and the deep ocean and processes across all sorts of time scales. It's a very, very powerful ratio, this Redfield ratio.